Today on Surfing the NASH Tsunami.
If we feed more data into the machine and bring it to the next level, we're discussing the first level, but I think we got to think beyond that. There's more the machine can do, and I think we'll see that emerge, and that'll bring us away from biopsy at the end of the day.
This weekend, over 200 NASH stakeholders have gathered in Deer Valley, Utah for the 5th NASH-TAG meeting. Join hepatology researcher and NASH-TAG co-director Dr. Stephen Harrison, liver wellness advocate Louise Campbell, Global Liver Institute president and CEO Donna Cryer, and pricing and forecasting guru Roger Green, along with our guest surfers, Dr. Jörn Schattenberg of the University of Mayence, Germany, and Dr. Naim Alkhouri of the Arizona Liver Health Institute, as they identify and debate major questions from NASH-TAG Session 2 on the Surfing the NASH Tsunami podcast.
For all those with an interest in NASH or more broadly, fatty liver disease, Surf's Up. Season 2, Episode 12, Session 2 of NASH-TAG 2021 starts now. It's now 6 in the morning, or slightly after that, in Utah. We went through one day of NASH-TAG, great day, I think, and the session starts again at 7:30 Utah time. So we're going to try to spend an hour getting our recording done now before the session starts. We will all listen to Session 3 then. We'll be back in the afternoon with a different cast of characters. Session 4, and then tomorrow morning we are going to sleep in and start at 7 o'clock in Utah time because there's no session after that. Today I've got Louise and Donna are here, and then we're fortunate to have with us Stephen. Hey Stephen, how are you this morning?
Doing great.
Okay, and Naim. Hey Naim, how are you?
Hey, good morning.
And Jörn Schattenberg, who is in Germany, not in Utah. Good morning, Jörn.
Hi, Roger. Hi, team.
We have an hour to get this done. I'm going to ask one question about the meeting, and then we're going to skip groundbreaker and kind of dive right into the topic. So let me start by asking Stephen and Naim, what does it feel like to be in the room at a virtual meeting with some socially distant attendance?
Thanks for that, Roger. So first of all, I have to say it's just terrific to be able to be around people, your colleagues, people that you've seen on Zoom or you've talked on the phone with or you've been distant from for over 15 months or at least a year. Our last NASH-TAG was January of last year, and for many people, including myself, that was the last time we actually were able to get together as peers and colleagues. So that was, 14 months ago, almost 15 months ago, just to be able to be around each other. And it's interesting, it was a little awkward at first because people didn't know how to act. Most everybody here is vaccinated, at least in the meeting, but still you were unsure, could you hug? Do you elbow bump? Do you fist bump? Do you shake hands? Do you take your mask off? Do you leave it on? And so that took part of the morning to kind of work through that. And then everybody fell right into a routine. And it was like we were back at an old meeting again, sitting around drinking coffee and having that interactive dialogue that really makes NASH-TAG so much fun. The downside to it is I can't talk to you guys necessarily face to face. Naim is here, but the rest of you aren't. And it's hard to have a discussion with somebody, again, kind of over a Zoom platform when there's so many people around that are in the meeting. So we miss that. We miss that camaraderie that comes with everybody being together, but I'll take it. I'll take the 40, 50, 60 people we have here in the room if that allows us to have a meeting in person and we can still virtually deliver the content in a way that is palpable and pleasing and understanding to the audience. I'm all in. Now, moving forward, I think the hope is we all get back together. We get the band back together. Everybody's here just like it normally is, but we kind of still have that virtual platform to allow others that couldn't make it for whatever reason. We still are able to deliver that content. So summary, I love it. I'm so glad I'm here. Wish you were here as well. I miss you guys very much and look forward to the next event. And I think that's what people are going to take away from this. They're going to walk away and say, you know what, that wasn't bad. We were able to do it. The airport wasn't that big of a deal. The car ride here wasn't that big of a deal. The meeting room wasn't that big of a deal. Maybe we too can have a meeting. And I think it's just going to begin to take off from here. That's my hope anyway. Naim, what do you think?
I couldn't agree more. I think it feels great to be here in person. As you said, as we approach each other initially, you wanted to check, have you been vaccinated? But luckily, almost everyone in the room has been vaccinated. So by the end of the yesterday we went back to normal. I think we have new CDC guidance saying that, for vaccinated people, you can, in a way, go back to the old ways and just, not have the mask if everyone around you is vaccinated. We are being abundantly cautious. We want to make sure that, we remain compliant with all the recommendations and we don't expose anyone. I think the virtual platform, it has a downside, but honestly, I like the access. I like that, we can reach more people, and hopefully this will help us make NASH-TAG even bigger next year. I think doing the hybrid meeting is a good idea in my mind. If we can just improve the way we interact with people attending virtually, that would make the meeting more valuable for them. But overall, great. I'm so happy I'm here. I'm so happy I can shake hands and talk to my colleagues in person and discuss ideas.
So Naim, how did all the skiing lessons work out for you?
I did not ski yesterday because I wanted to make sure I can do my session today. After that, I may go skiing, but I didn't want to break a leg. I'm a very amateur when it comes to skiing. I've done it 2 times in my life so far in preparation for NASH-TAG. So.
And Naim, he's done both of them in a place called the Arizona Ski Bowl. And if you know much about Arizona, it certainly isn't Utah. So Donna, Jörn, because you would have been there in person, as I would have, how did virtual feel to you?
Well, certainly it was, I missed the connection. I missed the people. I missed the, just leaning over and asking a question about what someone thought. But I really thought it was highly successful. The sound quality was clear. It was easy to get in and out of the platform. You could see the slides. And for those of us a little myopic, we could, enlarge the slides there. Although it's usually a small room, so you could easily see the slides even from the back. But it was nice to be able to look at the slides in detail. So I feel like I am smarter, as I always am, for having attended, and more informed from this really carefully curated current information on NASH. So I'm really grateful it was available.
Yeah, well, I've just punched in the distance, and I'm 5,190 miles away from you guys up there in Utah, Park City. So that is a distance, but it feels good to be online and discuss and see you and just hear, how Naim takes the questions, rebuttals. It's, you might know the data, but the discussion that evolves around the data is, of course, what really brings the knowledge or your perspective and the topic, and be able to participate over the distance from here, from Europe. That's really just something I enjoy, and I'm glad you guys went out there to have that discussion and and we can follow online.
Yeah, I enjoyed it. I thought it was very concise and it was nicely done. What was probably the most surreal moment was we'd done the recording with Minal, and the next minute she's on the TV. So it was bizarre to see people interacting that you've been interacting with over the recording of the podcast, and I enjoyed the format. I'm slightly closer than Jörn, but not too much closer.
My simple takeaway is that it was great to be there in person. I think the hybrid meeting was also really of value. It'll be interesting to figure out whether there's a way to bring some of the meeting to the people more effectively, to the people who are on the screen. This is an effort to do a little bit of that, and we'll see what we can come up with in the future, but great meeting. Stephen has to be in his car in 50 minutes to get over to the next session, so I'm going to skip completely groundbreakers and just get to the meat of what we wanted to talk about, which is basically to consider what we heard in yesterday afternoon's sessions, starting with platform trials and then going on through, I think, some really fascinating stuff through the reader reliability issues that Quentin laid out so neatly, and then to those presentations from PathAI and HistoIndex. I want to talk about what we learned, and really not just what we learned, but how we're processing things differently because of what we heard. So simple format, 3 questions. The coin toss says we start with whichever co-director or person in the room we have with us. The co-director would be Stephen right now. And 3 questions, really simple. One point or issue you heard during the session yesterday afternoon that you found novel or surprising, or you think is likely to drive a different course of action. And then there'll be 2 follow-up questions. What did the presentations and the session specifically tell you about this? And what are you taking away that you might do differently as a result of having listened to those 4 hours? And so, Stephen, let me start with you. One point or issue that you heard that I doubt surprising, but novel or likely to drive a different course of action.
Yeah, interestingly, the way I would answer that, I would answer the first 2 questions the same way, and the third question I would do something totally different. So first of all, the session 3, innovative trial design platform trials for NASH, fact or fancy, was really my first exposure to what this is. What is a platform trial? It's not something I'm familiar with in the U.S. and in the field of hepatology. It sounds like it's been done relatively frequently, or at least it's known to the oncology space and maybe some other disciplines in medicine. But for hepatology, it was novel for me. And I think, Nicholas and Vlad did a very good job of explaining it. I love the way Vlad broke it down into 2A, 2B, 3. He talked about what he liked about it. He talked about the potential opportunity for doing this in NASH. And then he highlighted the pros and the cons of each. And I think for me, we can talk about what I felt about it, I guess, in the second question. But to me, that was probably the most intriguing thing of the afternoon session. Having said that, I presented the seladelpar data, so that wasn't novel to me. But I do think it was important for the field to talk about this interface hepatitis and these plasma cells and even cholestatic disease that could be present in NASH at baseline. And it's interesting, when we look at drugs used to treat NASH, sometimes we see alkaline phosphatase drop. And obviously with the FXRs, it goes up. That's kind of a marker of how the drug's working. But when we see it drop, we often scratch our head and say, why is alkaline phosphatase dropping when you're improving insulin sensitivity or you're improving, inhibiting de novo lipogenesis or other improvements in liver cell health? Why are we seeing drops in alkaline phosphatase? I just can't help but wonder if maybe we're having an impact on some baseline cholestatic disease that really heretofore has not been something that's been routinely described. And then, we could talk for a whole hour on the digital path piece. And I really appreciated the roundtable discussion and Dave Kleiner's ability to process how he felt like that would be included or how he would utilize digital path. And then the comments by Andy and Dean and Quentin, I think, were very apropos. So it was hard for me to pick the one thing I thought was the most intriguing. But if I had to whittle it down, I would say that the idea of platform trials and how that would be done in a NASH setting was probably the most unique thing I took away from the afternoon.
Let me toss out a question first to you and to Naim and then to everybody else, since you guys basically run trials to some degree for a living. I know you do too, but in a university setting and in Europe. What do you think will happen in the States in the conduct of trials done by organizations like Summit or Arizona Liver Institute around the issue of platform trials?
I was intrigued by the idea, Roger, with the platform trials. I think, it has many potential for improvements on what we do in terms of having one placebo arm, maybe having a single screening procedure, maybe having the patients, allocated to another trial if they don't meet the criteria for one trial that you thought they will be a good candidate for as part of the platform protocol. I liked what Vlad showed in terms of the size of the placebo arm becoming smaller and smaller as you progress with the protocol. I noticed several issues that I think will probably slow down the implementation of this in the United States. Stephen mentioned that, for his patients, and I think it's the same for mine, that, the acceptance of the idea that they cannot decide on which trial they go on. I think that's going to be a difficult concept for them because we really spend a lot of time talking to them about the trial, what to expect, how the drug works, what we've seen in preliminary studies. So I think it's important for them to have that choice in a way. I think, it works probably better in cancer trials, especially when you design like a basket trial where you have a single therapy that you're assessing in multiple cancers as opposed to an umbrella trial where you have a single disease with multiple therapies. I think it might be a hard sell also for small biotechs when they have just one agent that they're trying and they're going to lose a lot of control with master protocol like this. But, I'm intrigued. I was also very happy to see, the progress that took place with the EU Pearl project and the timelines that they have. So, of course, we're open-minded and we want to advance the field, but, I can see the challenges as well.
Let me jump in here and just say that from a European perspective, I think some of the hurdles and some of the maybe pushbacks we get as European investigators for companies to come do trials over here is that part of the regulations are a little slower and you got to go to another authority to get approval. And I think that's a clear pro on the side of our site where you have a contract, you have ethical approval, and everything moves faster once you have agreed on a common platform. So for me, I think certain things are expedited. From the patient's perspective, I would say they are concerned about their liver health. And if I can offer liver health in a platform trial, I wouldn't be too concerned that they won't accept this because I might not be able to tell them about the concrete treatment arm up front. So I think on this side here, I really like it from the regulatory and the organizational procedural aspects.
Yeah, Roger, I was thinking about it when they're presenting. I mean, at, Arizona Liver Health, it's the same at Pinnacle. We are kind of running a master protocol because we have several trials. We look at the patients, to screen for one trial, but sometimes, they don't qualify. We look at other trials, they end up enrolling. So in a way, we are doing it in real life, but obviously it doesn't have the same, placebo arm. So we are missing a few pieces. But in reality, at any big NASH research place, you have several trials ongoing and you try to allocate the patients to this trial or that trial. But I think, again, a master protocol will have advantages, especially with regulators.
Naim, if I take those last two comments and I put them together, what I hear is in this States, the integrating role is actually played at the trial site, and the patient might feel more empowered because they know exactly what drug they're going on. In the European model, it's more centrally integrated, and the patient, Jörn, has to be more committed to liver health as compared to willing to do a trial, which strikes me as being a bit of a difference. I don't know what you guys think.
I mean, the other thing is the field is moving so fast that, the endpoints may change in a few years. We may have a standard of care that's approved, or would you just keep patients on placebo? Would you have to start the new standard of care? So these are all issues, to think about.
Let me just chime in. So first of all, I think Jörn makes a great point, because when we think about clinical trials, and I talk to sponsors about, okay, who we're going to bring into the trial, how many sites do we need, where are we going to get them from? And when we begin to think outside the U.S., it always comes down to the regulatory issues. How quickly is it going to take them to get up and running? And for the speed at which these sponsors want to move in the NASH space, they want to have these early phase 2As enrolled in a matter of a couple months, 2Bs enrolled in less than a year, phase 3s enrolled in less than 18 months. And often to get IRB approval in Europe, Jörn, you know this well, it's a while, sometimes many months. So these early phase trials are often enrolled before sites are up and running. So I think this idea of shared governance, central facilities, does give an opportunity for faster startups, lower cost, higher quality data, maybe even faster enrollment once it's done. But the issue here in the US, Naim hits on it. I have a panel of 5, 6, 10 protocols in front of me, and I look at a patient's demographics, I look at their medical history, I look at what ConMeds they're on, their FibroScan, their AST, whatever non-invasive test, MRE, whatever I have in front of me, and I try to match them up with the studies that they are most likely to qualify for, and then we move forward. The issue is, if they screen fail for whatever reason, I can't just put them in another study. I've got to screen fail them, I got to tell them they screen failed, then I've got to talk to them about another option, bring them in, screen them for that option. And Naim knows this as well as I do. We have people that will do that 2, 3, 4 times until we can get them into a study that they qualify for. So there is a benefit, I think, to having a menu that they could go into. But my concern is simply, my patients are savvy and they want to see the protocol ahead of time. They want to know what work has been done in a preclinical model, what issues came up. They want to know if there's any human data, what issues arose there, what the risk-benefit ratio for them being a part of that trial. And most of my patients take a lot of time to work through that, and they come in, and if I were to tell them, yeah, that's great, but I can't promise you you're going to get that. You may get this injectable over here, which has a completely different adverse event profile, but also a different efficacy profile. I can't see them routinely wanting to accept that. It's a novel idea in the field of liver disease and NASH. I just don't think it's ready for prime time yet. But we'll see what the EU Pearl, that organization does. If we can get some traction on it, I think we'll get experience with it, and maybe we'll, it'll be easier to adopt. But I think right now there's still significant challenges in the U.S. to have that.
Donna, Louise, Nick DeProspero had a pretty clear answer to that question about the patient, which is, he said, well, the patient could decide what to opt out of, just not what to opt into, right? Did that feel to you like a reasonably patient-savvy, patient-friendly response, or do you see that as being really different?
Well, I want to actually answer from the top, because I was on the board of the People-Centered Research Foundation that ran PCORnet. So 9 academic centers, 2 health plan networks. And one of the slides that was presented was from Janet Woodcock's articles, and all of those shared IRB, shared governance, all of those elements were her talking about PCORnet. So I have a lot of experience in trying to create this common governance. It is very difficult, but it can be done. And PCORnet was not set up for a specific type of research. It was about creating data linkages and a clinical trial, a national clinical trial infrastructure that could handle any type of research. And as I was tying it up with a bow and handing it back to Alkhouri, we stood up really quickly some COVID trials. And that was really exciting and really showed how all of these things we had put into place, because contracts with some of the most difficult institutions in the country were all done, in a matter of a week or two. All these things that we had built finally put into place, they were operationalized, and we were shown that use case of doing COVID research. So this is very attractive for all the reasons that it was described. The issue of can it work in NASH and can it work in the US? It seemed to me when I was listening to the EU Pearl, the thing that they were missing or could be the rate limiting factor was the recruitment of different sites to participate. And here what you've just heard Stephen and Naim describe is that in the US, we have this infrastructure of NASH, of several large NASH research networks. And so it's the flip side of the challenge. And so I would say that if we could, through the liver forum or through other mechanisms, find ways to streamline and I'm sure within each of your networks, that's why your networks are attractive, because you have streamlined the contracting, streamlined the IRB, streamlined all of these different things, and made yourselves easier to work with across a broader set of patients. If we can glean more of the benefits of this type of platform structure, building on the assets of what we have here, I think we can move a lot faster than the timeline of EU Pearl. And I think it would be really exciting to see. On the note of what patients would like to contribute to or what we would participate in, there are hundreds of NASH trials advising, and I do it from a sort of a 30,000 foot level, not associated with any one institution and any one particular trial, but advising patients on should you participate in NASH trials. I think the most important element, since we really don't know yet what works, so I can't say, well, if you go on this and it's a greater likelihood, I think it is an ethical place to talk to patients about helping us to advance the field, potentially finding something that will be beneficial for you, particularly if we have a mechanism to create shared placebo groups so more people end up in an active treatment of whatever kind. And I do think that the key choice for people is things like method of administration and just a few other factors that are key. But I think overall, I don't have a problem presenting to patients this concept of being part of NASH research writ large and helping us really quickly find the most valuable things and the things that work the best.
Okay, Louise, any thoughts to add on that?
I'll just add a thought, because on that question that you asked initially, I agree with Stephen. This was the section that I chose as the issue that was relevant. And it did get me starting the specific sort of questions about basket versus umbrella as the master protocol design, and particularly for NASH and NAFLD. As they're such heterogeneous diseases, both of these designs look to have benefits and drawbacks. But I started to think specifically on the basket design. Does this offer us now the opportunity to split the current trial outcomes? Because we tend to combine resolution of NASH with improvement of steatosis. Can we now, if you look at the basket design, split them out? Because we know some of the drugs are stronger in different components, and therefore look at them individually at different areas. Because Donna detailed last year that the ICD-10 codes are now including mild NAFLD, I think it was, and the lower grade. So this could offer design opportunities to develop the medications that have a greater effect on steatosis resolution, for example, to prevent the moving into F3, F4, which cause the mortality. So it just got me thinking in that way. But it also gave me the opportunity that when Manal talked last night again about non-disease progression being an outcome, again, to be able to target specifics with these populations rather than multiple outcomes at once, because not all NAFLD patients are the same. And I think it just comes with such different complexities that lots of other disease areas areas don't.
Louise, I had the same reaction thinking about what Manal had been saying yesterday afternoon as I was listening to that. I mean, and as Louise pointed out, it was bing, bing. We got done with Manal talking about non-progression as an endpoint, and then an hour later, this is going on, and she's in front of us running the session, and we're back in the same place again. Other thoughts on any of the questions around platform trials? I want to move on to another subject.
I think one thing I forgot to mention that patients always bring forward is the visit frequency. Most of our patients are working, maybe half or not that much, probably half are retired or working from home, so it's not as big a deal. But very often our patients will say, well, I can't do 2-week visits, or how many visits do I need to do up front before I get into a stable once a month, visit? Does it take me 30 minutes or an hour? And some of these protocols have pretty labor-intensive time requirements for our patients. Maybe they're doing PKs, right? So imagine if you got randomized to a trial that had 8-hour PKs. I don't have time to be there for 8 hours. I can come for 30 minutes. So you take that, significantly hamper enrollment in the U.S. if we were doing all of our enrollment off a platform trial, because we would be turning away at least half of our patients, in my opinion, just because they're not used to that idea. These patients are used to having a sheet of paper in their hand, they read through the protocol, and they make a decision. It's not this concept of, well, we want to help your liver, but we don't know exactly what you're going to get or how many visits you're going to do or whether you're doing PK or not PK or how, you're getting an MRI or you're getting a biopsy, but you're not getting a biopsy. I mean, they want to know the details of everything they're jumping in. And I think with what my understanding of a platform trial is that they're going to have to be more willing to just do research and hope that it works rather than specifically understanding exactly what they're involved with.
I think, though, the opportunity of some of the master protocols and is greater sort of standardization and smoothing out. And if we could make all the trials a little bit more patient-centric, then I think that's, I'll reveal my secret agenda to make all of the trials more patient-centered. So the workload is rational, or has a better chance of being rational and feasible for someone's life flow, as Mark Boutin would say, in any trial that they participate in.
Donna, that's a segue right into this whole notion of digital path and making life easier, from the concept of trying to work around the challenges of semi-quantitative assessment of collagen with light microscopy and all the variables in the NAFLD activity score. And the frustration there comes when I have a patient that I screen for a study. A pathologist says the NAFLD activity score is 6. You have 2 for ballooning, 2 for steatosis, and 2 for inflammation, but they were cirrhotic, so they don't qualify for that non-cirrhotic trial. So I pull them back, I screen them for another study that's non-cirrhotic, send it off to different pathologists, and they come back and say, yeah, this is great, they're stage 3 disease, but their NAS is 1, so they don't have a little fat, and that's all they have, so they don't qualify either. What do I tell my patient? Do you have NASH or do you not have NASH? The only thing that's clear is you have a lot of fibrosis, and I can't get you into a study. And I think this is where Digital Path steps in and kind of smooths out those rough edges. And I think Quentin Anstey's comments in his talk were eloquent, and I think they really drove home where AI is going to interface with our current pathologists to drive a clearer picture of what's happening in the liver. And then, in my opinion, that's going to transition us to a better understanding of the NITs that we're working on, right? Because now we're going to be comparing our non-invasive test to a digital path platform that has been looked at by a real live pathologist, and they concur with what the digital path is saying. But now we're able to adjudicate that non-invasive test against digital path, and that gets us to that level of sensitivity and specificity that the FDA really needs to transition to that non-invasive test as the endpoint. And I think there's so much to talk about with digital path. I want to make sure we hit on some of that in this podcast today, because really some phenomenal discussions were had that I think are going to drive this field forward coming out of that session.
I think, just to pick up one thing you were saying there about the potential for opportunities for platform trials, and I think we're learning through COVID that one of the opportunities for the platform trials was multiple different new units providing patients patients across different areas, which can increase the diversity of those patient populations, which is something we desperately need in NAFLD and NASH. But also, we're looking at ways that we can go out and fibroscan these patients within their community, making it patient-centric and combining taking of the bloods and doing those study visits to address exactly the issues that you're raising. That the more complicated a study is, the more we lose patients because we're working. Well, let's fibroscan you and take your bloods in the evening, or do it first thing in the morning before you go to work in a centre. So I think we are adapting techniques to be able to offer more patients the opportunity to participate in a greater way that's patient-centric and allows them to live their lives and give us the best data available.
And I think that can be done, that decentralization of trials, bringing trials into the community, into the home, hopefully will persist after COVID, and can be done in with a non-platform or non-platform trial. The decentralization and hybrid trial movement has accelerated to such an extent, and also has been acculturated within FDA now, providing those waivers, that there's so many active discussions about how to maintain that flexibility into the next years is exciting, certainly for patients and somebody who's worked on decentralization of trials for a very long time. I wanted to make the comment about the second part, that what struck me, because I'm always looking for challenges and barriers to adoption of new technology or new ideas, and I was struck by I wrote it as the plight and the pathos of the pathologist. So I heard Dr. Kleiner's heartfelt plea to not leave him behind, and I heard people's affirmations that AI, at least at this point, is meant to support the human pathologist. And I think that we need to recognize that we are causing a disruption, but it's disruption for a purpose. But I think we need to be mindful of those that we are disrupting and be thoughtful and mindful and intentional about carving out a new role for them so that AI can actually proceed apace. Because I feel like if we are not mindful, there will be a quiet or not so quiet, subtle or not so subtle pushback as people try to maintain their past value and contribution. And I think it's about finding new roles for people so that their expertise is honored, but the technology and its full potential can be realized.
As I was listening to that entire talk yesterday, the whole idea of the law of unintended consequences kept screaming through my brain. And you just hit on one side of that, which is progress gets pushback. And there's certainly massive potential pushback over the plight and pathos of the pathologist. But if you guys know, because we've all been hanging out enough, I immediately went to football. And where I went to was something called VAR, where they decided that referees made bad decisions, so they were going to have video help the quality of the decisions. And they've been doing this for 2 or 3 years all over Europe now. And the first thing you learn is it winds up getting used in ways no one anticipated. So they spend 5 minutes figuring out if somebody's shirt sleeve is offside, which will disallow a major goal in a match. And then you get all these people saying, well, this is no better. But frankly, if you took it away, then you would find out that you were missing it. And they're having a horrible time dealing with the unintended consequences of whatever path they take. Message being, Stephen, this is really exciting, but I think Donna does have a point, which is we have to proceed thoughtfully and with discipline, one step at a time, to make sure that everybody stays in the tent and that we are figuring out what the unintended consequences are as we go along before we've morphed into something we never meant to be exactly, and we're stuck sitting there.
I'm actually saying something different. I'm actually saying we need to move this really quickly, but we need to recognize that there are going to be very intentional consequences, because we are trying to replace, in a lot of ways, the pathologists. Be honest and transparent about it, but let's be intentional then about finding a new role and a way for them to honor their expertise so they don't block it. I'm saying move. I want it to move faster, but let's be smart and recognize what the barriers are.
We put the patient first, right? That's what we do. And I'm drawn to Quentin's conclusion slide where he says there are inherent flaws in semi-quantitative scores such as the NAS or SAF, nonlinear categorical descriptions of architectural changes without reference to quantitative changes in liver collagen, misclassification at the boundary between categories, inter and intra-observer variation in interpretation, potential insensitivity to therapeutic effects. These issues with an imperfect reference standard complicate development of better non-invasive testing biomarkers and hamper drug discovery pipelines and ultimately drugs for our patients. So we see these challenges and we see an opportunity opportunity to rise past these challenges. And I get that we have to be sensitive to every colleague that we work with, whether it's a radiologist, a pathologist, a endocrinologist, a hepatologist, a gastroenterologist, primary care, but the focus should be on our patients and what do we need to do to get good drugs in their hand. But before that, it may be equally important, make the right diagnosis. It's all about fibrosis. Fibrosis portends a worse prognosis. We need to make the right diagnosis to give our patients the right prognosis and get them on the right treatment. But all that requires a better understanding of what's happening holistically in a liver that unfortunately is heterogeneous in its disease severity from one segment to another, and trying to get at what that patient's disease state is challenging. And if we're seeing there are avenues that allow us to have a clearer understanding of that, I think we need to adopt that. Now, we need to do it carefully and thoughtfully. Yes. Do we need to consider everybody involved? Yes. And there is a way forward for pathologists, but we need to embrace some of this, I think, more than we're doing.
Stephen, I want to make a point about what you just said about making the correct diagnosis. And I want to actually talk about what you mentioned with the SELA-DELPAR program. I think that was a very important presentation. I deal with these issues on a daily basis with the fact that when you do a biopsy in someone with suspected NASH, NAFLD, often we get the pathology report and they say, oh, we see mild interface hepatitis. We see a few plasma cells. And I struggle with this with my nurse practitioners, sometimes if you have a fellow and they say, oh, well, it doesn't look like NASH because there's some interface hepatitis. You look at the whole picture in terms of immunoglobulins, autoantibodies. The patient is a, 45-year-old male with no autoimmune disease history. And it just, for me, obviously it is NASH and we say, no, it's okay. This is not autoimmune hepatitis. But the fact that, these features are being noticed on the biopsies as part of a NASH clinical trial, I think that's very important. The other piece about, the cholestatic features, and I see mild elevation in ALK-Phos commonly in my patients with NASH. You see ALK-Phos of 140, 150. As you said, with weight loss and sometimes, the treatments we use or being in a trial, you see a drop. And I think this speaks to the cholestatic features that we see. So I think this data is very valuable. I think, sometimes it actually confuses physicians, and many of these people end up having maybe a slightly positive SMA. They end up being started on steroids, and that will make their NASH worse. And I think knowing about these features is very important. In terms of Path AI, I think, the issue of semi-quantitative assessment is a big deal. What people don't realize, let's talk about steatosis, for example. If your pathologist gives you a score of 1 for steatosis because you have 33% steatosis on biopsy, if on repeat biopsy you go down to 7%, which is a significant drop, your score is still at 1. This is what we miss with the pathologist interpretation. And this is the issue when we are trying to validate Path AI against the pathologist, especially with the NAFLD activity score. I mean, when we say imperfect gold standard, it's a real issue. What I would like to see, honestly, with Path AI and similar technologies, HistoIndex, is take, biopsies from patients that were done 20 years ago, and then see what happened to these people longitudinally in terms of liver-related outcomes, mortality, liver cancer, and see if you can identify new features on the initial biopsy that actually predict outcomes. I mean, I realize there are some technical issues and, these technologies, you may not be able to apply all of them to older slides. But at the end of the day, the reason we care about NASH versus NAFLD, it's based on the fact that, when we reviewed older slides, we found that patients with NASH tend to progress more, to have more liver-related outcomes. But we might be missing a lot of things on the biopsy that an AI algorithm can pick on and help us predict, how the disease is going to progress. And the last point I want to make is, as a field, we really need to move away from histology completely. This is something we do now to validate new therapeutic targets. But, when you talk about a disease that affects 25% of the global population, histology has to be replaced by something that's easier to obtain, that doesn't have the risk of significant bleeding in 1 in 5,000 people, doesn't have the risk of dying after a liver biopsy. biopsy, I mean, this is real. I mean, the risk of dying after liver biopsy is 1 in 10,000. It's very low, but when you're talking about millions of people, it makes me cringe. So I think as a field, we need to move to something like a real liver function test, like pulmonary function test, a real, way to assess disease response to treatment like an A1C. We can't rely on histology moving forward. We are stuck with it for the next 5-6 years, but eventually we will move on. And, I've seen it with hepatitis C. I mean, I haven't biopsied a patient with hepatitis C in years now, and that used to be the standard of care. So I do believe that we will move completely from histology at some point in the future.
Roger, allow me to comment and pull these two things together quickly, what Naim and Stephen said. I think machine-augmented histology interpretation is here to stay. This is for sure. And I think I'd like to see next generation of machine augmentation and making the diagnosis. What Naim mentioned is very important, pulling together labs, having that alkyne phosphatase integrated into an algorithm that then together with histology gives a probable diagnosis. I'm sitting down with my liver pathologists every week to discuss the slides, and this is an advantage I have over the machine. But if we feed more data into the machine and bring it to the next level, we're discussing the first level, but I think we got to think beyond that. And there's more the machine can do. And I think we'll see that emerge, and that'll bring us away from biopsy at the end of the day.
Jörn, when I talked before about the law of unintended consequences, I think that the comment you just made is exactly where I'm going in Naim's comment about 20 years ago, which is let's make sure as we move this stuff going forward, and Donna, fast, slow in terms of weeks or months is less of interest to me, but a more thoughtful approach will decrease the probability that we're going to make errors in the next generation that we could have avoided if we took a more holistic approach to it, which is the thing I'd like to make sure we don't do. I'm back at, Louise is smiling, but I'm back at goals getting called for shirt sleeves, which nobody ever intended to do in the first place, but it's where that wound up because nobody got the rules right enough when they started. I just think it's worth time being a little more prudent. So, Stephen, let me ask you a question. The one most important thing that will be different in 2 years because of what we heard yesterday, or in 5 years because of what we heard yesterday. Quick 3-sentence answers. Stephen first.
I think if I blend what I presented from the lessons learned relative to the seladelpar hepatotoxicity review committee, where we have come to a consensus, at least among 3 expert pathologists, that there are additional lesions in NASH that we need to be aware of. I think that will be instrumental in helping us understand more about the natural history of this disease, as Naim was alluding to. If there's a lot of portal interface hepatitis in a baseline slide with associated additional NASH features and F2 disease, does that interface hepatitis link to a worse outcome? And this is where I think digital path will allow us to evaluate that on a much quicker platform. We'll be able to stain, if you use Path AI, you'll be able to stain for that and machine learn that much more quickly, I think, than routine path. If it's histoindex, you take an unstained slide and let the laser do the work on identifying it. And again, understanding how that's going to help us learn more about the natural history of this disease, I think, will be important. I don't think in the U.S. we'll have platform trials in NASH in the foreseeable future, but it's a dialogue I think that'll continue to build as we move forward with our meetings where we debate and discuss this. Jörn makes a fantastic point about opening up Europe to more opportunities for early phase trials with this platform, and maybe that's a way that we can work more collaboratively. But I hear the sensitivity of the pathologist in this whole thing. We're colleagues and friends with all of them, but I think 5 years from now, we need to be beyond semi-quantitative assessment of the features of NASH. And so that's what I'm taking away from the session yesterday.
Naim, biggest difference in 2 and 5 years?
Yeah, I mean, I'm excited to, learn more about platform trials. I think, I want to see how things shape up with implementation with the EU Pearl. I think we're all open-minded, and if we see that this is the way forward and it helps us with enrollment and patient satisfaction is actually higher, that we will adapt this model. So I think, who knows, maybe in 5 years that will be something that we do on a routine basis. Again, it's all about, acceptance by patients, sponsors, and the research side. rates. I think, histology is here to stay for the next few years, but I would love to move completely away from histology in the next 5 or 10 years. I think the way I look at Path AI, it's really to augment the pathologist reading and avoid some of the issues we're having now with the concordance, and helping the clinical trials also succeed by evaluating some of the features that are missed based on the NAFLD activity score and the definition of NASH resolution and fibrosis improvement. I learned a lot also from the histo index presentation about the different types of septa, whether it's progressive, regressive, broken, stable. I think these are important findings. I think, again, providing a quantitative assessment, especially, longitudinally, and how things change in response to treatment and being in a trial. So these are my takeaway from this session.
I very much echo Stephen and Naim. I think it's going to be interesting. I think the EU Pearl delivered the sort of master plan in about 2023, so it'll be very interesting. But I think the one thing that might be really nice about a platform trial for NASH is the potential to be able to reduce the age groups into the pediatric and adolescent population in a way that's safer, better followed, to be able to look at that natural history of the disease, but also to target a growing and concerning population in the development.
So when I think about the day as a whole, and particularly the afternoon, it seems that we're recognizing that NASH, both clinically and as a research field, is rife with complexity. And so whenever you have complexity, I think the solution is to put an overlay of simplicity around it. And so the themes of being able to have a trial process that will provide a standardized lifestyle intervention so that we can see more clearly a treatment effect, that there may be sort of one entry point into NASH trials, that they're more adaptive so that patients can be, assigned to different trials more quickly and be less likely to be exposed to a placebo group. And then, machine learning is applied more widely and will have had a role in validating non-invasive technologies. So I see this simplification over the very complications of NASH as being our way through, and being able to churn through more quickly and efficiently the number of drugs, the number of factors, the number of different patient profiles that are involved in here, to start having greater clarity on which drugs, which mechanisms, which combinations of mechanisms, which combinations of NITs, in which sequence we will need to use. I think we need to use a lot more technology at scale to be able to move people and through faster to be able to gain the knowledge that we need in the time that we need it.
Yeah, I think we'll see trial design change in terms of using more virtual aspects of safety assessment as far as that's possible, integration of multiple data points. And in the end, I think Donna highlighted it, it's about the patient and extent to which he is informed and able to choose and make a decision, an informed decision for himself, what type of trial or treatment he'd like to go on. So I think this is going the right way in terms of having more patient-centric view and aspect. And I'd like to support Naim in his plead for leaving histology behind as a part of NASH trial concepts here.
So my answer to that question, I want to use a phrase that Stephen used on podcast from time to time, #RealTalk. The transforming event for how we diagnose patients will, at the end of the day, be when drugs get approved. Because once drugs get approved, histology will not be a viable technique anymore, and we're going to have to use the best tools we have instead. So to me, things that came out of this meeting, Michael's comment in the opening session yesterday, the PDFF is valuable but not predictive in and of itself, coupled with all the challenges around semi-quantitative scoring and where we're going with digital, says to me that we have a couple years, maybe 3 years, to start to give doctors advice on how to diagnose and prescribe drugs to patients as these drugs become available. To me, that's the biggest change, and the thing I think we need to center towards as best we can. The platform trials are pivotal. The better use of data in clinical trials is, I think, a critical adjunct to what I just described. But that's the moment that the wave's going to hit, and we need to be ready for it. Now, you should know, listeners, that everyone else got off this call before I said that to go to their meeting. They will not have a chance to respond until the Monday night call. This afternoon, when we come back, we will have Michael, Charlton, and Ian Rowe with us, and then tomorrow morning, it will be 2/3 of this crew. No Stephen, no Louise, but Donna, Naim, and Jörn will be back. With that, let me thank Mike Wilson, who will spend the next 6 hours making this sound reasonable, and Eric Rounds so we'll spend the next few hours making sure that you guys know that it's here to listen to. And Paul will tell you for everything he does, Buzzsprout listeners and the organizers of NASH-TAG have let us be a part of it in this way. I had a fantastic time listening, and I expect to have a fantastic time again today. And if you're attending the conference, enjoy. If you're watching virtually, lots of good stuff coming up. See you later. Stay safe. Surf on. See you on the podcast. Bye-bye now.
You've been listening to the Surfing the NASH Tsunami coverage of NASH-TAG 2021. Have any questions for the surfers? You can post them on the Surfing the NASH Tsunami discussion group on LinkedIn or Facebook, or send them to questions@surfingnash.com, and we will attempt to answer before the end of our NASH-TG coverage. Thanks for listening. See you this evening for session number 3 on the Surfing the NASH Tsunami coverage of NASH-TAG 2021.